In this section, Autonomic Pharmacology I, we'll be primarily discussing the neuroanatomy of the autonomic nervous system and cholinergic signaling in that system. In the next section, entitled Autonomic Pharmacology, we'll be talking mostly about the cholinergic system. As you're aware, autonomic pharmacology is very important for the control of the autonomic nervous system that's responsible for automatic or unconscious bodily functions. Controls of this will regulate heart rate, peristalsis, sweating, and so forth. And this can be divided into parasympathetic and sympathetic nervous systems. Neurotransmitters in the autonomic nervous system include many, including acetylcholine and norepinephrine. Most direct transmission from the central nervous system uses acetylcholine, meaning presynaptic neurons will release acetylcholine onto a postsynaptic neuron. Postganglionic parasympathetic neurons will also use acetylcholine, but postganglionic sympathetic neurons will use norepinephrine, except certain sweat glands which will use acetylcholine. It's very important to remember the anatomy of the autonomic nervous system for step 1, and to remember which types of receptors are used at each synapse. This is a simplified diagram for the sympathetic nervous system as well as the parasympathetic nervous system. In the sympathetic nervous system, the preganglionic nerve has its cell body in the central nervous system, whether it's in the brainstem or the brain or in the spinal cord. The preganglionic neuron will release acetylcholine onto a postganglionic neuron that has its cell body in a ganglion, and that ganglion is outside the central nervous system. Acetylcholine is the neurotransmitter that's released each time. The receptors here on the postganglionic neurons are of the nicotinic type. These postganglionic neurons will usually, in the sympathetic nervous system, release norepinephrine onto the target organ. In sweat glands and the sympathetic nervous system, it's acetylcholine that's released. In the adrenal medulla, acetylcholine is released onto the chromaffin cells, which then release mostly epinephrine, but also norepinephrine, into the bloodstream. In the parasympathetic nervous system, we have a cell body that's located in the central nervous system of a preganglionic neuron, and this preganglionic neuron will release acetylcholine onto a postganglionic neuron. This ganglion is usually located in the target organ, and acetylcholine is then released onto that target organ, onto either the heart, smooth muscle, or other types of glands, and usually counteract the effects of the sympathetic nervous system. The neuroanatomy of the autonomic nervous system is very important to remember. Remember that in the sympathetic nervous system, you have a thoracolumbar distribution of the release of the neuron from the spinal cord. The cell body of these neurons is usually located in the brainstem, but it can also be located in the spinal cord. And these neurons are released from the spinal cord at the levels of the thoraco and lumbar spine. In the parasympathetic nervous system, you have a craniosacral distribution, meaning that the neurons are released from the central nervous system at the level of either the sacrum or the brainstem. And these neurons, such as cranial nerves 3, 7, 9, and 10, the vagus nerve, or cranial nerve 10 being the most important, they go on to innervate the entire body in terms of the parasympathetic nervous system. But the important thing to remember is that the nerve bodies will leave the central nervous system at either the level of the cranium, the cranial nerves, or the sacrum in terms of innervating the kidney, bladder, and sex organs. This is an overall diagram showing parasympathetic on the right. You'll go over this more in your neurophysiology lectures and neuroanatomy lectures. In general, when talking about adrenergic receptors, we can divide them up into alpha and beta receptors based on their structure, and also based upon the second messenger systems that they employ. Alpha receptors are then divided up into alpha 1 and alpha 2 receptors. Alpha 1 receptors are found in blood vessels mainly, and they, when activated, can cause vasoconstriction. They mainly are associated with G10 and G11, which activate phospholipase C. Alpha 2 receptors generally are associated with GI and GO, which are known to inhibit adenylyl cyclase and also inhibit calcium and potassium channels. This leads to decreased cyclic AMP production. Beta receptors are divided up into beta 1 and beta 2 receptors. Beta 2 receptors generally found in the lung. Beta 1 receptors are generally found in the heart. Beta 2 receptors are also found in skeletal muscle vasculature. These receptors generally are thought to stimulate GS, which stimulates adenylyl cyclase and calcium channels. Beta 3 receptor is generally found in adipose tissue and is responsible for metabolic process. The concept of dominant tone is very important. In a certain organ system, you will have a dominant tone, and the dominant tone of that organ system can either be sympathetic or parasympathetic. And each tissue is different. Giving medications such as ganglion blockers, like hexamethonium, trimethophan, or mecamylamine, will have a certain effect on the tissue, and their effect on that tissue will allow you to determine whether the dominant tone in that tissue is sympathetic or parasympathetic. These medications, the ganglion blockers, will block nicotinic cholinergic receptors at the junction, generally thought to be in the ganglion, the place where the presynaptic and the postsynaptic neurons will touch each other. These medications are not used anymore, but they can still be on the USMLE. Hexamethonium is different than the other two in that it's a non-competitive inhibitor of the nicotinic cholinergic receptor. The effects of the ganglionic blockers will differ based on the tissue. For example, ganglion blockers will cause vasodilation. This means that in vasculature, you have a dominant sympathetic tone, because sympathetic tone causes vasoconstriction, and it's being blocked by the ganglion blocker. They'll also cause an increased